we have a system now where we can clone temperature-sensitive alleles of all the essential genes onto a plasmid, and we can put them into a diploid that's deleted for the gene that's on the plasmid. We can convert that to a haploid where the essential gene's deleted, but the cells are alive because they have the TS allele on a plasmid. We can shift this strain to the restrictive temperature, find an extragenic suppressor, and then we can ask, can we kick out the plasmid? So can the suppressor actually bypass the deletion allele? And how many essential yeast genes in yeast can we delete and actually bypass with a suppressor? And so this, I like this one because I could bet my colleague, Fritz Roth, head-to-head, tete-a-tete, moi versus Dr. Fritz Roth, right? Yeah, head-to-head. So what did I bet Fritz? And Fritz is one of these guys that if you just lay a number out there, he'll go, I don't think so, right? He's a contrarian. So I laid out a number 15%. He goes, no way, all right? I think I bet him a bottle of wine. Yeah, it wasn't. Because, when someone does that, you start to second-guess yourself. And so Yolanda did this as a collaborative project with Fritz. So she, we got, 1,000 TS alleles covering, a lot of the essential genes. She gets a bunch of suppressors, and we're sequencing all these things now. And in the end, she could bypass 124 genes. And so guess who wins the bet? Yeah, so we're talking, right. And so that was fun. And the reason I made that bet was because the essential gene set is the same in Cerevisiae as it is in Pombe, almost identical, even though they are hundreds of millions of years divergent, and except for 15%. So there's 15% of the essential genes are different. And we can see that in fact, it's that set that is non-essential in Pombe that can be suppressed largely in Cerevisiae. So it kind of all fits. All right, so the last thing I'm just going to say is Elena has been looking at trigenic interactions, and it's the same game. But we took quantitative features of the digenic network to pick double mutant queries. So they either had high degree or low degree. They either had very similar profiles or weak profiles, which is a measure of functional similarity. The queries either showed a negative genetic interaction, a low one, or a high one. Again, the genes in the same pathway. If it's an essential pathway, often have a strong genetic interaction. Anyway, so she can map trigenic interactions, and they have the same properties as digenic interactions. They occur amongst genes in the same bioprocess. And I'll show you an example of one. So there's a trigenic interaction. All three genes are mutant. In the other cases here, it's either a single or a double mutant. And there's a little bit of action going on, but the trigenic is the one that falls apart. And when we look at these interactions, so the two query genes that we mutated are involved in secretion, and here's the double mutant query. It shows a bunch of secretion interactions, so these black and blue guys, but it branches out into DNA synthesis and repair, and this is often what we see, is that many of the interactions are connected to the function of the query genes, but the trigenics are a little broader in specificity. And so the bottom line is that the ones that trigenics are enriched for genes that have these particular properties on the digenic network. So they occur often at the level of complexes or pathways or within the same bioprocess. They often modify a previous digenic interaction, so the trigenic network is built up upon the digenic one. But the amazing thing is that while there's 500,000 digenic interactions, there's 100 million trigenic interactions in a yeast cell that we can predict. And so that when you get in the bioprocess there, if they don't show a digenic interaction, you can start combining two or three things, and you cause enough havoc in that bioprocess that you create this unexpected phenotype. And so we think that understanding the bioprocesses of human cells and then looking at variation in genes in the same bioprocess might allow us to start looking at inherited phenotypes. And so with that, I will just stop and then maybe have one question or something. Yes, a lot. You see aneuploidy a lot in suppression, and there's a fitness cost with aneuploidy cells. Don't like that, but when you go from a really sick cell, you can suppress it with aneuploidy. I don't think we've looked at that, but by the time we get it and sequence it, it should be pretty stable. But we've never tried to evolve beyond that. But yeah, you see, it definitely comes up all the time, and we'll see. We even have this one case where the genome, well, it's with the signal recognition part. So you take all the components of the signal recognition part, and you can delete them and bypass them. So there's some other way to live without it. And the way the cells do that is they duplicate the genome, and then they lose one chromosome. Yeah. So is there a way to estimate how often this interaction yields? Yeah, so they're definitely with suppression, because we look for suppression more than once. You can draw a graph and figure out whether you're saturated or not. We do have an estimate of false positive, false negative rate, which is surprisingly high. We buried that in the subdietary data, but it's around 30% or 30 or 40%. And yeah, I'm sure we can do a better job at it now if we look at it more carefully, but that's about how often we miss interactions. So
Einstein likes to say that allele-specific suppressors can be specific where you are the bypass suppressors to be less specific. For example, in a bacteria that if you have a where the mutation has a slow cell that suppress you can argue that that's really specific. That you've got a specific pathway.
I mean, most of the bypass guys are pretty specific, and partly because I don't think we're picking up weak suppressors. We were selected for pretty strong suppressors. But yeah, I don't know all the data, but yeah, Yelena's done all that analysis, and in general, they are very specific. Yeah, I know, it's just like that other thing we were talking about today on the train with the dots. He doesn't believe because he... So when they cluster together because they have a similar pattern of genetic interactions, then they're placed side by side on the map, and then we can go, these genes are annotated by SGD, and so it's independent functional annotation, and we can scan their annotations and see which ones are enriched in the clusters. Yeah, if they had used our data to create those functional annotations, it would be circular reasoning, but they don't use our data.
So from the 17 years of experiments with one condition, you said you started to evaluate how things are being open UI with each condition that you can
So what we've done is we've taken, we have those 1,000 genes that are in that hierarchy that behave really well in our assay. We now put those on an array, and then we cross in a bunch of genes from every bioprocess. So we make double mutants, and then we do that under a bunch of different conditions: salt, different carbon sources, drugs in the plate. And there's a few people who've done this out there, Trey Ideker, with DNA damage conditions, and they've really emphasized that the network changes quite a bit. They call it a flex network. But what we find is that the core network doesn't change at all. If you're a really solid genetic interaction under standard laboratory conditions, if you're a solid genetic interaction, even if there's DNA damaging agents in there, there's going to be some genes that maybe aren't even active under laboratory conditions that need to be induced or something if you had a DNA damaging agent, and those probably will show up for different genetic interactions, but they're not the majority. So the core network, just like the essential gene site, is highly conserved for superseding highly associated with the core network is going to be solid under different conditions, and there'll be a few genes that are changing.
Where something happens, the responses to stimuli, when it happens in the relative to other processes,
interactions that we are completely ignorant of the whole area of biology in the process. So the two talks the stories I'll be telling are one done by Dan, postdoctoral fellow at the University of Minnesota, whose expertise in Drosophila provided the opportunity of translating these imaging approaches, which we've done in living cell systems for many years, into live animals. And the other one, which is going to be our attempt at making some impact on human health, is in the area of developing drugs for rare cancer by Veronica Burns and Yunhui Cheng. My introduction, since I have two very different topics, I apologize, it's somewhat presumptuous, but bear with me. I'm going to start with asking a question which to me has been puzzling. I think most of us in this room who have been at our area of investigation for some time are proud of the advances that we made over the last 5 or 10 years. We think of things that happened 10 years ago, they were really hard, and now we can do things that are amazingly much more than we could 10 years ago. Now, if we compare with what we're doing in many basic sciences to what's happening in drug development, this is a plot of the number of drugs approved. Please apologize for rather ancient slide, but the picture hasn't improved. Despite enormous effort, as measured here by a number which is beyond my sort of comprehension in terms of financial impact, the success rate of drug development is woefully low. And the puzzle here is, why is this? We are making such enormous advances in science. Why is this not translated into... for inviting me, and it's great to be here in beautiful Paris. So I'm going to talk about this genetic landscape, and before I get going, I just want to say it's all a major collaboration with Brenda Andrews, who's the director of our institute here at the Donnelly Center. And I'm going to try and explain why this graph is a model of the cell. That's basically what I'm going to try and tell you in this moment. Yeah, it could be anything, right? But here's the model that we so we really don't have a good model for how they grow together. Systems biology tries to do that in a quantitative way. It's not that we're very good at it at this stage, but it's sort of the beginnings of it, and our project has kind of contributed to this. So I'm going to talk about budding yeast, which bunch of us work on here. David are leaders in the field, and it's a eukaryotic cell, so it's a billion years further developed beyond prokaryotes, and it's got a nucleus, a mitochondria with its own DNA. It's got all the goodies that we find in human cells, and so it's been a major organism. And in fact,
いらっしゃいませ。当店では店内奥のコミックコーナーにてセットコミックを販売しております。今話題の作品から懐かしの作品まで幅広く取り揃えております。表示価格から最大 50% オフと大変お得です。数量限定ですのでお見逃しなくセットコミックコーナーをご覧くださいませ。
いらっしゃいませ。本日もブックコンプラスオギックプロイ北口店にご来店いただきまして誠にありがとうございます。ご来店中のお客様にご案内申し上げます。当店では今週末の18日と19日の2日間、セットコミックが 20% オフとなるセールを開催いたします。セットコミックコーナーでは今人気の作品から表現作品やバック貸しのコミックまで幅広くご用意しております。また、メルティシールが貼ってある商品は今回さらに安くなり大変お買い得です。見逃しないこのチャンス、18日、19日はぜひ当店のセットコミックコーナーをご覧くださいませ。本日も当店ご来店くださいまして誠にありがとうございます。続きましてお客様の呼び出しを申し上げます。番号札5番のお客様、生産終了いたしますので入り口前、大体カウンターまでお越しくださいませ。